Welcome to the Deep Dive. Today we're doing a really fascinating exploration, diving deep into mitochondria.
Yeah, those tiny things inside our cells.
Exactly. And the material you sent over, especially, Nith Lane's book, Mitochondria and the Meaning of Life, it really highlights they're way more than just the powerhouses we learn about in school.
Oh, absolutely. They're central to, well, almost everything. Evolution, complexity, even why we have sexes, aging. It's all connected back to them.
So the plan today is to unpack that. We'll use Lane's work, those figures he includes on, like mitochondrial networks and lifespan.
Right, and the discussion's drawing on loads of research. We want to connect these, microscopic dots to the big picture of life.
We are getting bogged down in super technical jargon, hopefully.
Yeah, we'll aim for the aha moments, show how these tiny engines are tied to grand evolutionary stories and, well, our everyday lives.
Okay, let's get into it. So the sources mentioned our understanding wasn't always there. People first thought mitochondria did what? Made proteins or fats?
Something like that, yeah. Initial ideas were a bit off track. It took until, the mid-20th century? Folks like Kingbury, Kennedy, Leninger.
They nailed it down as respiration centers.
Exactly. But the origin story goes way, way further back. That's the really wild part.
You mean the endosymbiosis theory? Lynn Margulis?
Precisely. Back in the 60s, she really pushed this idea that mitochondria are basically ancient bacteria.
Bacteria that got swallowed.
By another cell.
Engulfed, yeah.
Yeah.
But not eaten. It was more like a merging, a symbiosis. This wasn't hostile. It was mutually beneficial.
Yeah.
And it fundamentally changed life on Earth.
So that double membrane they have, that's the evidence.
That's a big piece of it, yeah. It's like a fossil signature of that event. The inner membrane looks bacterial.
And the outer one, like the host cell that did the engulfing.
You got it. Plus, the actual stuff the membranes are made of, the lipids and proteins, also points back to bacteria.
Wow, so this one event, it basically allowed for complex life, eukaryotic cells. That's the prevailing view. It seems to have been maybe a unique event. All eukaryotes, that's us, plants, fungi, we either have mitochondria or show signs we used to.
Which is a huge difference from bacteria and archaea, right? They're simpler.
Much simpler generally. Smaller, no nucleus, no complex internal compartments, usually just a single circular chromosome.
Whereas eukaryotes have linear chromosomes, histones, all that complex organization.
And critically, thanks to mitochondria, a much more powerful way to generate energy.
Now, the sources mentioned a twist on this origin story.
Yeah.
The hydrogen hypothesis.
Ah, right. Martin and Miller's idea. It suggests the initial benefit wasn't necessarily about oxygen.
Oh, what then?
It proposes the bacterium, the protomitochondrion, might have been producing hydrogen,
Okay.
and the host was an archaeon, maybe a methanogen, that consumed hydrogen. So engulfing the bacterium gave the host a direct internal fuel source.
Huh. So the energy advantage came first, maybe even before oxygen respiration became the main thing. Interesting angle.
It's a compelling alternative view of those very first steps.
Okay, so shifting gears to how they actually make energy. It's not just like burning wood, is it? Lavoisier called it oxidation, but it's more controlled.
Exactly. It's not about releasing heat directly. It's a redox reaction, a controlled transfer of electrons, step by step.
Like a tiny electrical circuit.
In a way, yeah. That lets the cell capture the energy efficiently instead of just letting it go as heat.
And Warburg and Kylan,
Yeah.
they figured out the components involved.
Mm-hmm.
Respiratory pigments, the chain.
Right. Warburg indirectly identified key molecules needed for oxygen use. Kylan then conceptualized the respiratory chain, this series of protein complexes.
Cytochromes and stuff.
Yeah, passing electrons down the line like a baton race, ultimately to oxygen, forming water. It's this gradual energy release that's key.
And the Krebs cycle feeds into this, breaking down food.
Precisely. It's the central processing hub. It takes bits of food molecules, breaks them down further, and crucially, rips off high-energy electrons.
Which then get fit into that respiratory chain.
You got it.
And the whole point is making ATP. That's the goal.
That's the main energy currency, yes. Respiration makes way more ATP than older processes like fermentation.
Like a lot more.
Oh, yeah. Think maybe 30-something ATP per glucose molecule with respiration versus just 2 from fermentation. Huge difference. Engelhardt, Ochoa, they help show this link between oxygen respiration and massive ATP production.
The book made a point about the high-energy ATP bond being a bit of a misnomer, though.
Oh, yes. Good point. It's not that the bond itself is magically high energy, it's about the disequilibrium.
Disequilibrium.
Yeah, the cell uses energy from respiration to keep a high concentration of ATP relative to its discharged form, ADP. It's like keeping a spring tightly wound. The energy is in the wound state, maintained by constant input, not the spring material itself.
Okay, that makes more sense. It's about maintaining a potential difference.
Exactly, and Peter Mitchell figured out how that potential is converted, chemiosmosis.
Right. The proton gradient, that was revolutionary.
Totally. He proposed the respiratory chain doesn't just pass electrons, it pumps protons, hydrogen ions, across that inner mitochondrial membrane.
Creating a gradient, like water behind a dam.
Perfect analogy. You get more protons on one side than the other, creating both a concentration difference and an electrical charge difference. Potential energy store right there.
And then the protons flow back through, ATP synthase.
Yes. This incredible molecular machine. As protons flow back down their gradient through ATP synthase, it spins like a turbine, physically mashing ADP and phosphate together to make ATP.
Wow, a tiny biological motor.
It really is. And this basic principle using a proton gradient to make ATP is fundamental. It's used everywhere in life, not just mitochondria.
And bacteria use that gradient for other things too, like moving around.
Right. Homeostasis,
Yeah.
powering flagella. That's probably why mitochondria pump more protons than strictly needed just for ATP. The gradient is multi-purpose.
Okay, let's talk about the genes inside the mitochondria. They still have their own DNA, right? But not much.
Yeah, a tiny circular chromosome, very much like their bacterial ancestors. But most of the original bacterial genes got transferred to the cell nucleus over evolutionary time.
So why keep any genes in there? If most moved, why not all?
That's the million-dollar question. Still debated. One strong idea is local control. Many proteins made from mitochondrial genes are core components of the respiratory chain right there in the inner membrane.
So making them on-site allows for faster, maybe more precise control of energy production.
That's a leading hypothesis. Plus, maybe some of those proteins are just really hydrophobic or tricky to import back into the mitochondria if they were made outside in the cell cytoplasm. Could be logistical reasons, too.
And that mitochondrial DNA, it mutates faster than our nuclear DNA.
Significantly faster, yeah. Partly because it's right there in the thick of respiration, exposed to those damaging free radicals, the reactive oxygen species.
The byproducts of energy production.
Exactly. And mitochondria don't seem to have the same sophisticated DNA repair toolkit that the nucleus does, so damage accumulates more readily.
This all loops back to the evolution of complexity, doesn't it? The energy from mitochondria enabling eukaryotes to become, well, eukaryotic.
Absolutely fundamental. Think about all the things eukaryotes do that bacteria generally don't. Changing shape dramatically, engulfing things, phagocytosis, being much larger, having that complex nucleus with lots of DNA, developing sex, building multicellular bodies.
All that costs a ton of energy.
A ton. Mitochondria provided the energetic foundation that made all those innovations possible.
And the idea from Villi and Vita was that bacteria were stuck.
Yeah.
Limited by energy production on their outer surface.
Yeah, their argument is that as a bacterium gets bigger, its surface area for energy production doesn't keep up with its increasing volume that needs the energy. It creates an energy bottleneck.
But eukaryotes solved that by bringing the power plants inside.
Exactly. Internalizing energy production with mitochondria broke that surface area to volume constraint. It allowed cells to get much bigger, support a larger genome in a nucleus.
And that fueled the explosion of complexity we see in eukaryotes.
Precisely. More energy, bigger cells, more DNA, more potential for innovation.
Okay, now for a really curious connection. Mitochondria and the origin of two sexes. Why just two? The sources, Kirsten Hamilton, link this to mitochondria.
Yeah, it's a classic evolutionary puzzle. Why not 3 sexes or 10? Wouldn't that make finding a mate easier?
Seems logical, yeah.
But Hurst and Hamilton proposed that having just 2 sexes helps enforce uniparental inheritance of mitochondria.
Meaning you only get them from one parent, usually the mother in animals.
Right. And why is that important? To avoid conflict?
Conflict between mitochondria.
Yeah, imagine getting mitochondria from both parents.
Right.
You might have different mitochondrial lineages within your cell's heteroclasmy. These might compete or just not work well together, messing up respiration.
So ensuring they all come from one source keeps the mitochondrial population uniform and working efficiently. Like having one set of engine blueprints, not 2 conflicting ones.
That's a good way to put it. It prevents internal genomic warfare that could drag down the cell's energy production. So uniparental inheritance is strongly favored.
And typically the egg provides the mitochondria, sperm don't.
Generally, yes. The egg is huge compared to the sperm, packed with mitochondria. Sperm mitochondria are often in the tail, which gets discarded, or they're actively destroyed after fertilization. Different mechanisms, same outcome.
This connects to tracing human ancestry too, right?
Yeah.
The out-of-Africa stuff using mitochondrial DNA.
Exactly. Stone King, Wilson's work in the 80s was huge because mtDNA is mostly passed down maternally without mixing like nuclear DNA.
You can trace direct female lineages way back.
Right. By comparing mtDNA variations across global populations, they found the deepest branches trace back to Africa, supporting the idea that modern humans originated there relatively recently.
And that high mutation rate you mentioned earlier acts like a clock for this.
It's used as a molecular clock, yeah, to estimate when lineages diverged, but as the book notes, it's not perfect.
Oh, how so?
Well, we now know there can be rare instances of mitochondrial recombination, mixing of mtDNA, and natural selection definitely acts on mitochondrial genes too. It's not just a neutral clock ticking away.
So the picture's a bit more complex than just straight maternal inheritance and random mutation.
It is. There's this idea of dual control, the need for the mitochondrial genome to work well with the nuclear genome. They have to be compatible for optimal respiration.
Ah, so selection favors combinations of nuclear and mitochondrial genes that cooperate efficiently.
Exactly. That puts strong pressure back on maintaining uniparental inheritance most of the time to ensure that well-matched set isn't broken up every generation.
Okay, fascinating. Let's shift to probably the most personal connection for many people. Mitochondria and aging. The free radical theory is the famous one.
Right. Harman's idea later expanded. Basically, respiration isn't perfectly efficient. It leaks reactive oxygen species free radicals.
And these damage stuff. DNA, proteins, membranes.
Yeah, they cause oxidative stress. The theory is this damage accumulates over time, leading to the functional decline we call aging.
And there's that correlation. Faster metabolism often means shorter lifespan, like mice versus elephants.
Generally, yes. But there are wrinkles. Birds, for instance.
Right. High metabolism for flight, but often surprisingly long lives for their size.
Exactly. And the exercise paradox, athletes have very high oxygen turnover, high metabolism during exercise, but aren't necessarily aging faster, maybe even the opposite.
So free radicals are likely part of the story, but maybe not the whole story, or at least not in a simple linear way.
Seems that way. There are other theories, like Metawar's idea about late-acting harmful mutations accumulating.
Because selection doesn't care much after reproduction.
Right. But that doesn't fully explain how flexible aging can be, how diet or genetics can change lifespan. And while specific mitochondrial mutations do cause diseases, the overall load in normal aging might be less than expected by the simplest free radical models.
What about this retrograde response mentioned in the reading? Mitochondria talking back to the nucleus.
Yeah, that's a key concept now. When mitochondria are stressed or damaged, they send signals out to the nucleus.
Like an SOS signal.
Sort of. It tells the nucleus, hey, things aren't great down here. And the nucleus responds by changing gene expression, maybe boosting antioxidant defenses, altering metabolism, trying to cope. It's a communication loop.
So they're not just passive victims of damage.
No, they're actively involved in the cell's stress response.
Yeah.
And another crucial mitochondrial role in aging is apoptosis.
Programmed cell death. How does that fit in?
Mitochondria are central regulators of apoptosis. They can trigger the cell death cascade. One idea is there's a metabolic threshold.
Based on ATP levels.
Yeah. If mitochondrial function drops so low that ATP production falls below a critical point, the cell might initiate apoptosis. It's a quality control mechanism.
To get rid of badly damaged cells before they cause bigger problems.
Exactly. But the downside is, if this happens in tissues with cells that don't easily replace themselves, like neurons or heart muscle,
You lose function over time. That sounds like age-related decline.
it's thought to be a major contributor, yes. The loss of irreplaceable cells via mitochondrially controlled apoptosis.
It's quite a thought. The organelles giving us life also hold the keys to our decline and death. The book suggested almost all aging factors funnel through mitochondria.
That's the provocative idea, yes. That whether it's genetics, diet, toxins, their ultimate impact on aging and age-related disease is mediated or calibrated by how they affect mitochondrial function, that metabolic threshold, and apoptosis signals.
Because energy and stress response are so central.
Right. And we do see respiratory chain problems and free radical leakage getting worse in aging tissues or diseased organs. It suggests mitochondria are a central node where all these aging pathways converge.
So, wrapping up this deep dive, looking back at the huge timescale, What are the biggest evolutionary legacies of mitochondria, according to the sources?
Well, the book really hammers home three massive ones. First, the origin of the complex eukaryotic cell itself. Couldn't have happened without them.
The foundation for all plants, animals, fungi?
Everything complex, yeah. Second, the origin of two sexes, driven by that need for uniparental inheritance to keep mitochondria happy.
A very counterintuitive link.
It really is. And third, the phenomenon of aging and programmed death as we see it in complex organisms, deeply tied to mitochondrial health and function over a lifetime.
So the key takeaway really is, wow, mitochondria aren't just background power packs. They're fundamental players, architects of complexity, shapers of sex, mediators of aging.
Absolutely. From that initial ancient merger billions of years ago, they've been co-piloting the evolution of complex life. Their influence is just immense, woven into the very fabric of what we are.
Which leaves us with a really big question, doesn't it? If they're so central, what else don't we know? What might future discoveries reveal?
Exactly. And could we, learn to intervene? Could manipulating mitochondrial function be a route to tackling aging or treating diseases in ways we haven't even conceived of yet?
The potential seems huge, if incredibly complex.
Definitely complex, but understanding mitochondria better seems key to understanding life itself at some very profound levels.
Well, we've definitely only scratched the surface here. It's an amazing topic. We hope everyone listening feels inspired to look deeper into endosymbiosis, mitochondrial genetics, aging.
Yeah, there's so much more there. It really is a journey into the foundations of life.
Thank you for joining us on this deep dive. It's been truly enlightening.